Okay, we'll begin the second session today. I'm Izumi Ozawa. I'll be chairing this session. The first speaker of this session is Dr. Hidehiko Komatsu of National Institute of Physiological Sciences. And in the interest of time, the introduction is on the symposium brochure. So please look at that. Please begin.
Thank you, Chairman. So first of all, I should express my sincere gratitude to all participants. So the title of my talk is the neural representation of growth in visual cortex. But as first speaker from the Shitsukan group, I think I should explain what Shitsukan means, though Nishida-san has already given some explanation. So what is Shitsukan? Shitsukan is a Japanese word to indicate perception of materials and surface qualities of objects, as well as sensory impression of their fineness. And Shitsukan consists of two Chinese characters, shitsu and kan. And shitsu means quality, and kan means sense. So this is a very convenient word, so please remember Shitsukan. Okay, so growth is a very important attribute affecting impression surface and also Shitsukan perception. So though understanding neural mechanisms may not be elegant science, but we are interested in the neural mechanisms of growth perception. So this slide shows the monkey brain, and visual cortex is indicated by this shaded area. And visual cortex consists of many visual areas, and this slide shows the connections between different visual areas. There are two major streams of information flow in the cortex. And there are two major streams. One is called dorsal pathway, and the other is called ventral pathway. And both pathways originate from the same area that is called primary visual cortex, or V1. And this slide shows the connection between different areas. And the dorsal pathway goes from V1 to the posterior parietal cortex and is related to space and motion perception. And ventral pathway goes from V1 to the inferior temporal cortex, or IT cortex, and is related to object recognition. So the question is, how is growth information represented in the visual cortex? So in this talk, I talk about some of studies which studied the, how the information related to growth is represented in the visual cortex. So to start with, we conducted an experiment, a functional MRI experiment using monkeys. So to search for the areas responsive to growth stimuli. So the monkeys were sitting in a bore of the MRI machine like this and faced the tangent screen display. And monkeys conducted visual fixation task. And these are the stimuli presented to the monkeys. So this is a glossy object. This is a matte object. And these are phase scramble version of the glossy and matte object. So we searched for the area. So these stimuli are presented one by one to the monkeys. And we measured brain activity. And we searched for the area where the activity to glossy stimulus is stronger than any other three stimuli. So this shows the result. So orange color indicates a region where the activity to glossy stimulus is stronger than any other three stimuli in the previous slide. So orange color covers the region from V1 to some part in the inferior temporal cortex along the ventral visual pathway. So this slide shows the organization of the ventral visual pathway. It starts from V1 and includes areas like V2, V4, and the inferior temporal cortex, or IT cortex. IT is divided in three sectors: posterior IT, PIT, central IT, CIT, and anterior IT, AIT. So our functional MRI study has shown that glossy and matte surfaces are distinguished in the areas in the ventral visual pathway. So the other studies, previous studies have shown that higher areas in the ventral visual pathway are also related to another important aspect of Shitsukan perception, that is material perception, like Kant and Guttel, or Kabina, Pratesi et al., and Hiramatsu et al. In humans, or Goda et al. In monkeys. And also, Ed Conner and colleagues have found neurons selective for 3D shape. That is another important object attribute in the inferior temporal cortex, especially in the lower bank of superior temporal sulcus, STS. So we thought this area should be a very good candidate area to search for neurons which distinguish a variety of growth. So we conducted neural recording experiment from this area to look for the neurons which distinguish a variety of growth. So how should we make stimuli to test the growth selectivity? So this slide explains two major reflection components. One is diffuse reflection that reflect light evenly in any directions. And another component is called specular reflection that reflect light in strongly to one particular direction. And this slide shows, this is a Roland paper, the 3 physical parameters related to growth. So one is diffuse reflectance, low D. Second is specular reflectance, low S. And third is spread of specular reflection due to surface roughness, alpha. So diffuse reflectance influences on, diffuse reflectance indicate the strength of diffuse reflection and influences on the overall intensity of reflected light. And specular reflectance indicates the strength of specular reflection and influences on the intensity of highlight. And roughness is the surface roughness at the microscopic level and influences on the spread or blur of highlights. So to test the selectivity of neurons to various growths, we generated stimuli with various combinations of these 3 parameters. To generate a stimuli to test growth selectivity of neurons, with regard to the surface reflectance, we picked up 33 surface reflectances from MAR BR database. And this shows the parameter low D, low S, alpha in previous slide of these 33 stimuli. And with regard to the object shape, we generated 10 these object shapes. And with regard to illumination, we picked up natural illumination from the direct light probe gallery. And by combining these 3 factors, we rendered object images using Radiance software. And this slide shows an example of stimulus set, 33 stimuli, which was used to test growth selectivity of neurons. So these stimuli consist of a variety of growths, and these are presented one by one to the monkey, and we tested neural responses. And the recording site is, as I said, a lower bank of the supratemporal sulcus in central IT, CIT. And we found that this shows the results. There exist neurons in the STS that selectively respond to a specific range of growth. So this slide shows 2 examples which we call growth selective neurons. And this slide shows 2 examples of growth selective neurons. So in this figure, the response magnitude is indicated by the size of icons. So this neuron strongly responded to a shiny object with sharp highlight. On the other hand, this neuron responded to glossy object with blurred highlight. So we found many such growth selective neurons in this area. And this figure shows the distribution of response magnitude in the parameter space shown in previous slide. So low D, low S, and alpha. So this neuron strongly responded to stimuli with large specular reflectance low S and small alpha roughness. But this neuron strongly responded to stimuli with large specular reflectance and large roughness. So then we asked how a variety of growth is represented by the population of such growth-selective neurons. So if these neurons are related to the growth, we can expect that population of neurons similarly respond to a pair of stimuli, which if they have similar growth appearance. But if they have dissimilar appearance, the response pattern should be different. And we examined this by computing cross correlation coefficient between all pairs of 33 stimuli. So this shows two examples. So in this figure, this graph, the horizontal axis indicates a response of population of 57 growth-selective neurons. So each point indicates one neuron, and to this stimulus. And vertical axis indicates the response of the same set of neurons to this stimulus. So though these two stimuli have different color, they have similar growth appearance. Both have a shiny object with sharp highlights. And the correlation is very high. So this indicates that population of growth-selective neurons behave similarly to this pair of stimuli. However, in this graph, both stimuli have red color, but growth appearance is very different. This is matte and this is shiny. And in this case, correlation is very low. So we can continue to analyze representation of growth due to population activity by computing the correlation coefficient and 1 minus correlation coefficient as a neural distance for each pair of stimulus, and make distance matrix for all pair of 33 stimuli, and then apply multi-dimensional scaling analysis. And this slide shows two-dimensional embedding of a variety of growths based on the activities of population of growth-selective neurons. So in this plane, the distance between two stimuli reflects the difference in the activities of population of growth-selective neurons. So as you can see, stimuli with similar growth appearance are located nearby. So here, you can see object with large specular reflectance and small roughness. In other words, shiny object with sharp highlights are clustered here. And here, object with large specular reflectance and large roughness. Those objects with broad highlights are clustered here. An object with large diffuse reflectance and small specular reflectance. In other words, matte objects are clustered here. So this result indicates that a variety of growths are represented systematically by the activities of population of growth-selective neurons in this area. Then the next question is, what kind of information about growth is represented by the activities of these neurons or encoded by these neurons? Because higher visual areas is thought to be related to visual perception, we thought these neurons carry information, encode information related to growth perception, or in other words, parameters related to growth. So previously, Pelacini, Capital already mentioned about this work. Pelacini, Forward, and Greenberg previously conducted psychological experiment to determine perceptual growth space. And they identified two axes, C and D, that characterize this perceptual growth space. And C, these are the two parameters. C is nonlinear combination of low S and low D, low S specular reflectance and diffuse reflectance. D is 1 minus alpha. So we can regard these as perceptual growth parameters. And in the next experiment, we made stimuli with various combination of C and D and tested the activity of growth-selective neurons. As it is also known that surface albedo or diffuse reflectance influence on growth perception, we generated a set of stimuli with different diffuse reflectance and tested neural responses using these stimulus. We found that growth selective neurons show clear selectivity in this perceptual growth space. So this slide shows 3 examples of growth selective neurons in this CD plane. And so also we found that neurons are tuned to particular direction in this CD plane. So this neuron are tuned along the axis. This neuron tuned along the direction close to C axis. This neuron tuned along in intermediate direction. Also, it seems that the responses seems to be represented by linear combination of C and D. So we try to fit the neural responses by this equation. So this figure shows the relationship between the actual response indicated by circle and the response fitted by this equation indicated by asterisk, and the magenta indicates a fitted plane. So most of the neurons are pretty well fitted by this equation. And also, from this fitting plane, we can determine quantitatively the tuning direction of each neuron as a direction of maximum slope of this fitted plane. And we found one interesting thing about the distribution of tuning directions. This slide shows the distribution of tuning directions of the growth selective neurons in the CD plane. So there's clear bias toward upper right direction. Roughly speaking, this is the direction to which perceived growth increases. Because there seems to be linear relationship between neural activities and perceptual parameter growth, we attempted to estimate each of the perceived parameters from the neural responses. And if this is possible, for example, C can be represented by something like this. However, large number of neurons cause overfitting, so we first compressed the neural activity by principal component analysis to 4 dimensions, and then applied multiple regression analysis. Then we asked, can the growth parameters be estimated from the population activities of growth selective neurons? We found the answer was yes. And each of the perceptual parameters are fairly precisely estimated from the population activities of growth selective neurons. So this slide shows this is a fitting for C, and this is a fitting for D dash. That is slightly modified version of D. And this is cubic root of rho D, fitting for cubic root of rho D. And in all cases, the fitting was fairly precise. And this slide shows the relationship between the actual value indicated by blue color and the estimated value indicated by red color for physical parameters, alpha, rho S, rho D, and perceptual parameters, D dash C, cubic root of rho D. And as you can see, the fitting is much better for perceptual parameters rather than the physical parameters. So this indicates that population activities of growth selective neurons seems to encode perceptual parameters rather than physical parameters related to growth. Then these experiments have shown that activity of growth selective neurons located in this area, CIT, seems to encode perceptual parameters like these. Then the next important question is, how is the selectivity to growth selective neurons formed in the visual cortex? But the short answer to this important question is no, we don't know yet. However, to obtain some hint on this important question, we recently conducted anatomical experiment to know from where this region where growth selective neurons are located receive input, the visual cortex, area visual cortex. So we injected retrograde tracer into the area in CIT where we recorded growth selective neurons. And this slide shows schematically the concept of retrograde tracer injection experiment. So this is the target area. In this case, this region where we found growth-selective neurons, and retrograde tracer are injected this to target region by something like microsyringe. And then the molecule of the retrograde tracer is taken up into the, from the axon terminal of the neuron, which send axon to this target region. And then the molecule is transported backward along the axon to the cell body, and then accumulate in the cell body. So by visualizing cell bodies, we can know from where this target region received input. And we found that retrograde level neurons were distributed some locations in PIT and some locations in V4. Of these, we are particularly interested in the region near slightly dorsal to the anterior end of posterior middle temporal sulcus, PMTS. So this slide shows the section at this level. And these dots indicate the retrograde level neurons. This is STS and this is PMTS, anterior end of PMTS. And in this picture, these brown dots are the retrograde level neurons. And there's a cluster of level neurons at the location slightly dorsal to the anterior end of PMTS. The reason why we are interested in this particular location is that previous studies have reported neurons sensitive to local luminance contrast in this region. This is a figure taken from the paper by Keiji Tanaka and colleagues in 1992. So they penetrate the electrode into the location here that is slightly dorsal to the anterior end of middle temporal sulcus. This is very close to the location where we found retrograde cluster of retrograde level neurons in previous slide. And this figure shows the optimal feature of neurons recorded at different depths along this single penetration. Most of neurons are sensitive to the local luminance contrast. Also, as you remember, these are the perceptual growth parameters, C and D, and of these, C indicates local luminance contrast around the highlights. And also, we have previously found that neurons sensitive to local luminance contrast are clustered in the same area, slightly dorsal to the anterior end of the posterior middle temporal sulcus, like this. So each circle indicates a tuning of orientation of local luminance contrast. So although it's highly speculative right now, we speculate that neurons sensitive to local luminance contrast in this area in PIT may provide information related to C to growth selective neurons located more anteriorly in CIT. Okay. To summarize, information related to growth is mainly processed along the ventral visual pathway. And growth selective neurons that can distinguish a variety of growth exist in the STS of the inferior temporal cortex, a higher area of the ventral visual pathway. And growth selectivity of these neurons are stable across 3D shape and illumination, and they may be extracting image features essential for coding growth. So I didn't talk about this result today. And population of growth selective neurons seem to encode parameters closely related to growth parameters, C, D dash, and cubic root of rho D. And we speculate local features related to growth should be detected in early visual areas, and they are integrated in extracellular areas, including portions in PIT. Okay. So this slide shows collaborators who participate in the experiment which I talked today. Akiko Nishio recorded growth selective neurons from CIT, and Nakazu Goda and Goki Okazawa conducted functional MRI experiment, and Takeaki Shimokawa conducted important mathematical analysis of data. And Noritaka Ichinohe collaborated with anatomical experiment and Takbano and Masaria participated in the experiment to record neurons sensitive to local luminance contrast. Thank you for your attention.
Thank you very much. We have time for comments and questions. Anybody?
So, beautiful work. And it occurred to me when I was listening, have you ever considered the possibility that neurons in this area might also be sensitive to dynamic aspects of gloss? You know, what people might call shimmer, or you know, that kind of extremely fast change of contrast through time.
Actually, we don't have I don't have no direct answer. I haven't tested dynamic stimuli. I suspect that they are detected in different areas, like I mean, more motion sensitive area, like you know, MT, MST detect motion signals, and then integrated in somewhere like MST or FST. And recently, Doshina and the colleagues have shown reported very interesting paper about what kind of flow is important for the dynamic aspect of growth. And they then at some more anterior stage, maybe they both static and dynamic things are integrated. Also, one recent study have shown that there's also other dynamic, but stereo or is also related to, in some aspects, related to growth. And the recent human functional MRI study have shown that in human, more dorsal area also, like V3A-B, involved in the growth perception. And they may be integrated in somewhere different, but I suspect CIT may be more static image feature.
Actually, my question was quite related to Ed's. So in the first one, when you were showing the fMRI, you had your stimuli actually dynamic. And there's this work by Katja Dorschner that she showed in Germany, where if you stick the features, the gloss features, onto the shape, it actually doesn't look glossy anymore. I wondered if you'd tried that as a control.
That is very important control. But the purpose we use dynamic image in that particular experiment is to enhance the impression of growth. So we could actually use a static image in that experiment. But if we test the dynamic aspect of growth perception, we certainly need that kind of control, I think.
So because that stimulus sort of equates for these local gloss features that are being fed. Of course, then you need motion. And interestingly, it's when the gloss features aren't moving and the rest of the object is moving that you get the gloss.
Any other questions?
So one very important aspect of material perception is the presence of invariance across other factors. And I know that you used a series of different shapes, but I wasn't quite clear from the analysis to what extent these neurons demonstrate invariance. And particularly, I was a little bit, so there's this very striking finding of the tuning, predominance of tuning towards the diagonal within your parameter space, but that seems to be strongly associated with local contrasts. And so if you could demonstrate that these neurons have a degree of contrast invariance, but are still tuned to specific regions of the material space, that would be stronger evidence, I think, for a material-specific response.
That is a very important question. About first point, shape invariance, I didn't show the data, but in the previous paper, we showed that we used the different shapes, and the selectivity, the definition of for the classification of the criteria for classification of growth selectivity in our study is that they have shape invariance, in the sense that they show the correlation of growth selectivity is significant. So, but many neurons showed change in the response magnitude, as you can control. And so that also means that because if we use different shape, the pattern, local pattern of highlight changes. So that is one control. But also, but as you said, we cannot deny the possibility that they are sensitive to local contrast. Actually, it's very hard to dissociate. But the best control is something like Bart has done. And so, yeah, we did some try, but not systematically so far. So, yeah, in future.
Well, that was my question. So.
Yeah, that's, that's the most important.
You saw me armed with the mic, so.
Yeah.
Anybody? I've got one question. Can you define sort of sharpness of tuning in this, say, for example, like a CD space? Or, I mean, did you examine, you represented each neuron as a single point in that space, but...
CD space, there are something like plane in CD space.
Oh, okay. Right, but, you know, yeah, I mean, cell 1 seems to be more specifically tuned than cell 3, for example, you know, in terms of the area that occupies in that space.
Yeah.
And my question is that, can neuron tune sharply to all dimensions, including the shape dimension? In other words, did you see any sort of a tendency where if you find a neuron to be relatively broad in tuning in CD space, for example, is it more sharply tuned, you know, in an opposite way for shape, or did you, I mean, you probably can get only impressions like that.
Yeah, and the general impression is that, as I said, it's like a plane, and so it's, this is, yeah, it seems to be a bit non-linear, but more, generally speaking, it's linear. So in that sense, it's gradual change in this space.
Anybody? Okay, thank you very much.